Alright, so this is the last regular lecture. We still have some more labs to do, I know, but this is the last of the video lectures you're going to have to look that's substantial like this. And so the last topic we're going to touch on is biophotonics. And the image here, this is, as you can see, it's a cross-section of an umbrella plant with UV-induced fluorescence of things that are already fluorescing in there. So you can see the different cell types and chlorophyll and things like that. So this is some of the imaging techniques we're going to be looking at today and understanding how they work. As we get started with this last lecture, I want to say nice work this semester. You've done a lot of hard work to get to this point. At the beginning of the course, I told you up front it was going to be a lot of work and then it was going to be a soft landing, sort of. And that's where we are. So I don't have a whole lot of new technical content to present at this point because you've learned everything up from the basics of how light propagates even through more advanced optical systems like interferometers. And so you really have a lot of the basics to understand optics, and that was our goal this semester. So in these past few weeks you get to apply it to some more applied topics like biophotonics, and then you get to do your own design lab. But again, there's not a whole lot of deep technical content to cover, and this should be all fairly easy. And so today's stuff, again, it's biophotonics. We're going to cover all of these topics from imaging to 96 well plates, flow cytometry, laser surgery, pulse ox, and capnography. And they're going to touch on pretty much all aspects of optics as we wrap this up. So the first one is fluorescence imaging. If you want to do simple fluorescence imaging, then what you do is you basically have a setup that's like this, okay? And you put your specimen down here, which could be, in this case, it's a dividing human cancer cell. And in this setup, we're just going to look at one type of fluorescence color. We're going to assume that this cell will fluorescence green, and then we can see what the cell shape looks like under this microscope. Okay? So the first thing I have is an excitation source. So it could be a bright lamp. Then I have an excitation filter, which only lets the blue light in. The blue light is then reflected off this dichroic mirror, comes down through the microscope objective. The blue light has high energy, causes the cell to fluoresce, and I'll talk about how you can make the cell fluoresce in a second. Sometimes you add a fluorescent dye. And then the green light comes back and it goes through the dichroic mirror, whereas the blue is reflected, and the green comes on through here to the detector at this point. Okay, so this is your camera system. So you only see the green light, so only the parts of the shell that you label will fluoresce. So for example, if you looked at this and used this setup to see this cell here, there are types of fluorescent dyes that you can add to the cells that only show you specific proteins or parts of cells so you can see and examine them individually. And the beauty of this is all this bright blue light coming in, none of it's going to wash out your detector. So you can basically have a very, very high sensitive detector, so it only has to see the light that's fluorescing because that's a really low signal, and all this excitation signal won't come back and wash out the detector or damage it. So what about this dichroic mirror? What is this? How do you think you make it? Well, here's a dichroic mirror you could purchase from Thor Labs. Look at the transmission and reflection. Reflection is very high here in this case for blue, and then it drops down into 0 in the green. Transmission very low for blue because it's all reflecting, and it transmits like crazy as we get over into the green and the red. So how did they make this thing? Well, look at this. You can see there's a strong bandpass. This is just like when we simulated those photonic crystals, those 1D photonic crystals with the multiple alternating layers of something like silicon dioxide, titanium dioxide, etc. That's exactly what they do. And because it's based on optical interference, you'll get strong transmission for one set of wavelengths, whereas another set of wavelengths will have strong reflection instead. And so in this case, all the wavelengths are reflected. So hence you get a dichroic mirror, hence the term dichroic, meaning two colors, one that passes, one that doesn't pass. So how do you make these cells fluoresce where you want them to? Well, you need to choose dyes for fluorescence. That involves a couple things. One is specificity. You choose a dye that binds to the protein or the cell wall or whatever you want to see of interest. So you can buy these proteins and they'll go and they'll stick to the right part of the cell you want. So for example, here's one from Life Technologies, not cheap, okay, $350 for 5 milligrams. Here's the absorption and the emission spectrum. And this particular one is used to basically stain G-actin and F-actin which are structural proteins of the cells. And it's very selective for that, meaning it will only have an affinity for those, and no other parts of the cells will show up. And so you can start to see cell structure and health this way. So you need that specificity, and they make different fluorescent dyes for different parts of cells, and they have ones that can target cancer cells versus regular cells and things like that. Second thing is quantum yield. You basically want to have as many of the photons that you shine on it and that are absorbed turn into photons that are emitted. So a high quantum yield is good, meaning efficiently fluorescing. And the other thing you want is a strong absorption cross-section at the excitation wavelength. So, you need the light to be absorbed, and so you want a strong absorption. And so absorption cross-section is different than absorption coefficient. We had talked about this for Beer-Lambert law, remember? Where alpha is units of 1 over centimeter, and it goes up here in the exponent when you look at the intensity, how it falls off with distance into a sample due to absorption. Well, to get absorption cross-section, you need alpha as well, but you multiply that by the atomic density, okay? And that's basically the atomic density of the fluorescent dye itself. So that gives you a measure of absorption cross-section, which means for a given amount of something, how strongly does it absorb. Now, if you want to figure out, like, how do I even do this? When you go to these companies, they give you great procedures. So they tell you, like, here's the chemicals you have to purchase, here's how you have to mix things up, temperatures, etc., and then you're ready to go at the end. Okay, and so they give you pretty good directions on how you can use these things for medical imaging. So if fluorescence imaging is all this great, why do we not do fluorescence imaging deep within the body? Why do we got to take the cells out, put them on a microscope slide, and then look them under a microscope? Well, there's two reasons. One has to do with light loss, the other has to do with light refraction. So the first one have to do with light loss has to do with two things. One, you get absorption by oxygenated and deoxygenated hemoglobin in your blood. That's what makes your blood red or blue colored, right? And if you look at it, as you go into the visible spectrum, it gets stronger and stronger. So this really wipes out the visible spectrum. You've got a lot of absorption here. And so you can't get light deep into the body because of this. So you say, well, heck, let's just go out here to the infrared. Well, when you go out into the infrared, look at the absorption spectra of water. It starts shooting up as well. So hemoglobin knocks out the visible. Then you go into the infrared and you lose as well. And of course you can't go deep into the ultraviolet because that absorbs like crazy. And UV light, right? Too much UV light gives you skin cancer, right? So you don't want to use that either. So these two work against you. And the last thing that really hurts you is tissues and cells. They scatter light. So just like a piece of paper, anything that's got multiple refractive surfaces inside of it doesn't allow a nice transmission, rather, and it scatters and reflects light. So that makes things challenging as well. So let's say we're coming back to the microscope setup and we would say, what's the smallest thing I could see? We never really talked about this in this class, but we did talk about it a little bit when we talked about how small you could focus a laser beam down. Remember, this was the diffraction limit in terms of how small you could focus a laser beam. And so the smallest D naught you could achieve was related to the divergence. So if I had a large divergence, meaning I really had a large angle at which I'm bringing this light down, I could get a smaller D naught because this is constant. This is wavelength here, right? Well, if you want to get a large angle here, like not like this, where I'm bringing the light in from a really large angle like this, how do I do that? Well, you need lens systems with a large numerical aperture. Here's a small numerical aperture, larger, larger. You can see now you've got that wide angle, which will increase theta, allow you to decrease D because this is a constant. And again, remember numerical aperture, you could look at it as light gathering power, but you could also look at it in terms of this equation and the minimum D naught you can see as well. And so if you have a larger numerical aperture, alpha is really large, the gathering cone of light. So get a lens system with a large numerical aperture, but what's that tell us about this D naught? Well, at the end of the day, you're going to be left with a resolution limit. So here's an example, 50x Nikon objective with a numerical aperture of 0.8, which is pretty good, and red light could barely resolve objects as close as 406 nanometers. So they need to be a little bit more than 400 nanometers apart to be able to see them. And that was determined, I calculated that, using this equation where we've got lambda, which is units of distance, which gives us our nanometers, 2 over numerical aperture. Numerical aperture is unitless, okay? So you can get numerical aperture from here, sub it in, and I got my wavelength put it in. That gives me the resolution limit. And so when you try to look at really small things, imagine if I have some little fluorescent things that I want to look at. This gives you this is a representation of the fluorescence intensity versus distance. So the peaks would be the most fluorescent spots. This is the top view, this is an angled view. And so you'll notice that each of these dots, when you go to really high resolution, all of a sudden you start to see these circular things around them called Airy disks and Airy patterns. What are those? Well, at this point you're seeing your diffraction limited, right? You see the same kind of things you would expect from a diffracting system. You're getting down to the diffraction limit where all of a sudden you start to see these diffractive effects start to appear up. So again, the main point, this is the resolution limit, and it makes sense because it comes back to the same principles we had talked about before for beam divergence and minimum beam waste D0. Now, in terms of this fluorescence imaging, sometimes you don't want to look at something small, but you just want to look at the total bulk fluorescence signal coming from a solution with cells or proteins or something in it. And so one way you do this is a microtiter or micro well plate, typically a 96 well plate. And what you do is you basically have all these wells, again, typically 96 of them, and you'll do something like, okay, I'll change pH this way, and I'll change chemical A in this way, and in all these I'll put some kind of, some kind of protein or something. I want to see how that changes, how it could fluoresce or something that would happen to it, right, or detect something. So I can do a lot of experiments at one time because I will image the fluorescence from all of these at one time. And the most powerful version of this is the enzyme-linked immunoabsorbent assay, or ELISA. That's basically the basis of most medical diagnostic testing in humans and animals. And so how's this work? Well, you put this well plate right here. Here's your microplate here, and you put the little fluids in all these little wells. Here's your excitation source. You've got a lamp. It goes through this tuning fork, which basically allows the signal to be chopped. That way, if you time the chopping with this detector array, you can determine which of the light is coming from here through the excitation, and then use AC filtering to remove out all the background light you might have. You've got some lenses and a filter wheel to get the right color for excitation here. It goes into a fiber distributor, and then you've got light coming through these lenses, so it shines down to each of these. The lenses then focus it down onto a detector board which has 96 photodiodes here. So you can use this to get very high sensitivity and rapid parallel imaging of fluorescence intensity. Recently, Dr. On here at UC, his company released a product in 2011. Just Google Cylome Optimax and you'll probably see it come up, that basically makes some substantial improvements on this technique using some clever microfluidics. So even more powerful is a technique called flow cytometry. It's a technique for analyzing and counting a large population of fluorescently labeled cells in a fluid stream. So put that fluorescent dye that sticks to certain type of cells and then starts to count them. Okay, so you might get a dye for cancer cells and a dye that sticks on regular cells, and then try to spot the ones that are cancerous and determine if you can count if someone's got cancer, and if they do have cancer, how many cancerous cells are in their blood or in tissue. So here's a flow cytometer. Basically, you say you draw a little bit of blood, you put it will be injected here, and then this is water flowing on either side that helps focus down this sample down to a single file lane of cells or particles that you're trying to see. It doesn't have to be cells, it could be even particles coming through here. Okay? And then you have a laser which can hit the cells, and there's two things you have here. You have a forward scattering detecting photodiode, and then you have this light coming out here to all these PMTs, photomultiplier tubes. Okay? So the forward scatter is the amount of light that if it hits a cell, you can imagine light could come in and be reflected here, right? And so that means that if it hits a cell, then the amount of light getting to the detector could decrease. So you could measure that. If you were like kind of looking at this, you'd be like, okay, here's my optical signal. Then it dropped down because there was a cell, etc. So every time the light intensity went down versus time, that meant it was getting in the way of the beam path. The reason you can use a photodiode for this is that you have a laser, plenty of optical power, right? But how much light can you get to fluoresce off a cell? Not very much, so that's why you need these photomultiplier tubes. So this way what happens is if the blue light causes the cell to fluoresce. And remember, you can stain regular cells maybe green and cancer cells as yellow or red, okay? Regular cell comes in, fluoresces green, green light goes to this PMT, and it says, I just counted a regular cell. And PMTs can be really fast too. They can detect very quickly as well. And regular cells are coming through, then a cancer cell comes through, and let's say it's stained red. Red fluorescence comes in, it just counts that it just found one cancer cell, for example. Okay? And so you need these photomultiplier tubes because they have such low light sensitivity that they can do the counting you need. Now look at these dichroic mirrors. See these DM, dichroic mirrors, meaning this one reflects only blue, lets everything else through. This one reflects green, lets everything else through. These are those thin film-based mirrors that pass some wavelengths but very high efficiently, but then block others as well. So at the end of the day, what you do is you look at this forward scattering photodiode signal and the side scatter, and that helps you, in addition to basically just counting based on the colors you see. So here's the greens, here's the red, here's the blues. You can also get ideas of things such as size. So things that are smaller or bigger could have different amounts of forward scattering, and so you could start to get ideas sizes and a little bit, and then you can do counts based on the detectors, blue, red, or green, that are counting different types, neutrophils, monocytes, or lymphocytes. And this is just a little bit in terms of how you get a nice column flow of single cells there. You need to have a low Reynolds number. If you take Dr. Papowski's class, he'll talk more about that. So the flow cytometries are very fast in throughput, up to 100,000 counts per second. It's amazing with commercial systems. And you can also sort things. If you count them, you can sort them too. So, for example, here's your flow of cells coming in. Here's your laser. Counts a red cell, sends it in the photomultiplier tube to a computer, electrical wire, and then when the cell gets to the point where it's going to be ejected as a droplet, you could put positive or negative voltage here to give the droplet a positive charge, no charge, or a negative charge. These charged droplets come down, then you have this electric field, which can then deflect them and sort them into different vials. And so you could do this to isolate cells to be able to examine them further. So for example, if I wanted to look at different types of cells, here's erythrocytes 10 to the 6 per microliter. Circulating tumor cells, which are cancer cells found in blood, are not 10 to the 6 per microliter, they're only 10 to the 2nd per liter. That's a huge difference in concentration. And so systems like this are great because then you can sort these down into as much as you can and then concentrate them and look at them, because you'll never be able to see this in a background full of these other cells if you want to do fluorescence imaging and things like that. So at that point, do a little bit of review, take a break, and again, hey, we're almost there on these lectures, so hang in there.